Answering MCAT questions in under 60 seconds. Researchers isolate a protein that runs as a single band on a non-reducing SDS page gel, but has two distinct bands on a reducing gel. The protein is most likely a what? So remember, when you're doing SDS page, you're separating proteins based on mass. If it's a non-reducing gel, that means you're not eliminating any of the disulfide bonds in that structure, okay? And so in the first, when it's non-reducing, you just get one protein. Okay. However, or one band, excuse me. But when it was a reducing gel, you actually reduced those disulfide bonds and you got two bands. So that means the protein had to have been a quaternary structure which was held together by disulfide bonds. And when you reduce those disulfide bonds, you break apart the multimer into individual monomers and you get two distinct bands. Okay. So if we look at these answer choices, A says the protein was most likely a heteromultimer that formed in the oxidizing environment of the endoplasmic reticulum. So in this case, in order for it to have been a multimer held together by disulfide bonds, you would need an oxidizing environment. So that checks out. But is it a heteromultimer or is it a homomultimer like the other answer choices? A heteromultimer is one where the subunits, the individual monomers, are distinct from each other, right? Whereas a homomultimer, all the subunits are the same. So in this case, it would actually have to be a heteromultimer. And why is that? Well, they said that the two bands that formed, or rather they said that after the reducing agent was added, two bands formed, meaning that because there is separation based on mass, each of the proteins forming those respective bands had different masses. So they were different monomers, and therefore it has to be a heteromultimer, not a homomultimer. And therefore A here is going to be the right answer, okay? Now B says heteromultimer that formed in the reducing environment. Well, reducing environments would not promote disulfide bond formation, so that's wrong. And then C and D are wrong because it could not have been a homomultimer. If it was a homomultimer, what would have happened is that after the reducing agent was added, you would still only get one band, except it would correspond to a lower molecular weight of protein, right? But you wouldn't get two distinct bands because it would still be the same molecular mass since all of the multimers are since all of the monomers are the same size, okay? The band would just be further down the gel because it is lower molecular mass, okay, than the quaternary structure. So A is the best answer here.